Okay, I'm not going into details, but this is a research proposal, materials and methods that we require. That section I'm discussing right here. The first thing in here is the cell survival technique. The MTDSS, crystal violet staining. There are some basic type of technique that's being utilized for cell survival or the cell line survival purpose to know how much your LC50, some kind of calculation like calculation like LC50. Okay, and the best way is your annex. I'm not sorry for cancer. It's annexin V. The fast technique, the flow cytometry technique being utilized. Trypan blow assay is a little bit older method which is not being properly considering nowadays. Forget about this. The in this picture here it shows the the migration of the cells when particular chemicals being applied. So it to shows that whether a cell line, whether it's moving towards that chemicals, or it's get affected by the chemicals, whether it gets synchronized or it gets moving towards anything else. For example, if we put some matrigel in a cancer cell, producing a producing a cancer cell, by giving the matrigel, we can clump those cancer cells at a particular site in a very proper shape. But if we don't apply the material and they simply put some cancer cells inside the region of insertion, then it will be bulge in a scatter way, and that's not good. So this is kind of thing. So during chemotherapy, what do we use? We use some chemicals before the surgery or after the surgery, or both in case of some case of cess also. So in the places where we use before surgery, that means the drug actually do in that region, in that particular case, that drug is preventing the proliferation or movement of any or scattering away of the tumor or cancer cell. Rather, it helps to get clumped, to get come together, helps those cells to get come together in a particular region, so that it will be easy to remove those cells, tumor cells, from that region. And in those cases where some chemicals are applied after removing the tumor, or after done of the surgery, to prevent the migration or mobilization of cell, like your most fibroblast cell, which actually clips those adhering cells and move spread away. So to prevent those kind of things, we use some kind of chemicals after surgery, and those chemicals also kill those sensitive cells, because it compared to normal cell, the cancer cells are very highly sensitive, so it will a little bit easier to kill those cells without affecting the normal cell line or cell, sorry. So this is important in that aspect. Here nothing is important. Here you can see crystal violet staining. In this, like 96 well or something, well plate having some oval shape furrow, where you put your desired drugs and some cell lines, and within some time interval, and check out within about 1 day or 1 hour, some different type of time interval. You can check this out whether your drug is actually working or killing. So by this method you can calculate your LC50 or something. That means how much your drugs should be administered in that particular cell line. So what should you consider as accurate amount of your drug. Then here in the structure is given in a round structure. It shows that different region you have placed your cell line, scattered away individually, and they group together, clumps together. And you can check this out with some drug effectiveness, etc. In this case you can see here you can see actually, for example, if this one is your cancer cell, and here is the cytotoxic T cell. Then the binding between this receptor with that of its protein on its surface can be controlled for can be taken as a consideration for immunohistochemistry assay or immunotechnic assay. We can use inhibitors to prevent this binding of that receptor with that of its protein, or we can simply just cleave out this region on the surface of its protein by breaking some bonds or adding something else. So this way we can inhibit anything else. Okay, in vivo condition, when we have our drug in your hand, we can use this to transfer, to check whether this drug is actually working in different organism or it's limited to the cell lines. So syngenic method, where the syn means similar, genic means genetic constitution. So if for example you consider a mice, then you put the drug inside the mice and then you clone you clone another mice from that cell line of this the previously taken mouse. So in this case, you can administrate the drug in that clone mice to check the activity or immune activity or immune response to that drug. This is your syngenic model. In xenograft model, what you actually use, you use nude mice. That means the nude mice do not have any immunity in itself. That means it don't have any thymus gland. The thymus gland is being removed, so it is very sensitive to its surrounding and very susceptible to various kind of bacterial viral infection. So they should be taken very carefully, and any tumor cancer cell could be easily administered inside this organism and could be possibly tested. Then another thing is that in orthogenic model and subcutaneous is a mode of introduction or introduce this is the way we can introduce our tumor or cancer cell line inside a tissue sample inside or in vivo condition. That means inside, for example, in mice pancreatic cancer, we have to cut the pancreas and then put inside completely inside in between the organ, that is pancreas organ, and that tissue, pancreatic cancer tissue, then after putting inside we have to stitch this organ and then we cover, and this way we can do the orthotropic model. But this kind of technique is very complicated and very what we call riskier as compared to that subcutaneous mode of infection, where we just simply place our desired tumor or cancer cell on the surface of that organ. Then the real-time PCR to know the up and down regulation of a different protein sample. For example, if you consider as a GPCR, G protein coupled receptor, as your protein, whether to see whether in presence of a drug your gene is expressing, this protein is being expressed as up regulated or down regulated. In that case, what you have to do, you have to pick your consider your cancer or tumor cell, and with the application of that drug and a control that without having that drug administrated. So two cell lines you have to pick up. One is a control, other one is drug administrated, and then you have to pick up the total mRNA from there, make it complementary DNA. Then what you have to put your primer, the forward and reverse primer, which is very specific to that gene, and then you can place it all those complementary DNA to this PCR, real-time PCR. And as you can see, the primer has only binding affinity towards its respective complementary DNA, so it only goes and bind to the GPCR related GPCR protein gene sequence, and that will be what we call it, that it will replicated, and by this we can know that whether your gene or respective gene for the GPCR is upregulated or downregulated in presence of that your drug, and you can compare with that of your control one, that means in absence of your drug. That's it. And some computational bioinformatics, like your RNA sequencing you can also do, and that will be really very important for your impact factor of your publication. And that really helps because there you can simply compare your RNA in presence of a drug and in absence of drug, and by that RNA concentration you can derive the genes which are responsible for that cancer, the drug administration effectiveness on regulating those reactions. So that's important, and that's really a little bit costly, but that really helps you bring your data sufficiently accurately. And other thing like your green process protein, which helps you to localize, to find the localization of your protein, of your desired protein. But in case of luciferase, it will helps you to show the expression of your desired gene. That's it, man. Thank you.